Hello, this is Michael Edwards, CEO at Bioinfo Solutions. This is part 3 in a video series I am doing on crowdsourcing coronavirus data. In the previous video, I explained how we collected gene expression data on the lungs response to SARS-CoV infection in monkey and mouse animal models. In this section, I will present the results of that effort and explain the metadata that is available for download. To access these files, go to my GitHub account at the URL down below, or find this link in the description for this video. For this example, I will describe the Day 1 post-infection dataset, but the format will be the same for all the other time points. I am starting off with the first tab of the Excel file labeled SARS-CoV Day 1 post-infection lung metasignature, which contains all the sample information and metadata for this time point. The individual SARS-CoV animal studies are given on the rows, while the variables are in the columns. Columns A through Q are all the experimental variables. The first column gives our ID for this entry, while the next column gives a description of that comparison used in that entry. Our next variable is tissue, and all results were derived from lung tissue, which contains many cell types like epithelial cells and immune cells, among others. We used two kinds of animal models, with the species and/or strain information given to the right. All the monkeys that were used were female, but I could not find any information on the gender for the mouse studies. We had a variety of animals with different ages, with the macaque monkeys at 8 to 15 years of age, which would make them about middle age for this species, and mice that were 52 weeks old, which would be considered very old for that group, a group that was 20 weeks old, which would probably be considered middle-aged, and a group that was under 8 weeks old, which can be considered young for mice. All the changes in gene expression were compared to the same baseline, which was non-infected lung, with all other variables for that animal being the same for that comparison. There were 7 different viral strains used in these studies, with the SARS-CoV MA15 strain being the most common. All the mouse studies used the same infection dose of 100,000 PFUs, or plaque-forming units, except for one study I could not get any information on. Monkeys are significantly larger than mice, so they required a higher initial viral concentration. All samples for these studies were taken at Day 1 post-infection, and all gene expression was measured using microarray technology, with all the monkey studies and one mouse study using Affymetric gene chips, and the rest using the Agilent technology. We also provide the title for the study entry with its associated gene expression omnibus ID number to the right. We drew comparisons from 6 different GEO entries from 3 different lab groups for this analysis. And finally, we also provide the year that these studies were registered with GEO. The remaining columns to the right contain the scale data on the magnitude of change for that gene to infection compared to normal lung. The gene symbols are in the header. These values are based on an Illumina scoring system that ranks the genes within each bioset based on the magnitude of their change. The gene with the greatest change in gene expression compared to the controls is designated 100, and everything else is scaled down to 0 from that. The base-based correlation engine platform we collected these data from only allows you to download the top 5,000 genes based on the sum of the Illumina scores across all datasets. We modified these scores to reflect the direction of change so that all Illumina scores signifying a change in expression below control levels were made negative, while all genes with an increase in expression remain positive. As an example, you can see this first gene here, CMPK2. Based on the Illumina scores, we can tell that the expression of the magnitude of change was very, very high compared to all the other genes in these data sets. Not only that, but we can also tell that this went up or increased compared to our control levels. If these were negative, this would mean that there was a high magnitude of change below our control levels. It is this scaling system that allows us to compare all of these results in the same data set. This tab on this Excel is also saved as a tab delineated text file in the same GitHub directory that will allow you to upload this into analytical software like ClueCore, Partex Genomics Suite, or even JUMP. The second tab is all the experimental variables in one place, and which is basically the information I just gave you. I know some people like to keep their sample information separate from the data matrix. This is the same variables I went over in the first tab. This last tab gives all the gene information for this meta-analysis, with the genes now in the rows and the biosets given in the columns. On this table, I also indicate the specificity for the change in this gene, with the amount of biosets out of a total of 13 where we observed a change in gene expression compared to the controls. And right next to that would be the percentage corresponding to the total amount of these biosets. I also include the overall gene score that gives the sum of the absolute values for all the Illumina scores for that gene, which again is used to determine the top 5,000 genes for download from a base-based correlation engine. The column next to it is the sum of the adjusted Illumina scores that now takes into account whether that gene increased or decreased compared to the control. If the absolute score and the score sum are different, that means all the gene expression for that gene was not consistent for all the samples in its direction of change, like this gene here. That shows an increase in expression in all but one of the biosets given in this data set. The highlighted column is the average of the Illumina scores across all datasets for that gene. In future videos, we will use this average values as our standard to put together a cumulative signature of the host response at day 1 after infection that we can upload into various bioinformatic programs to gain biological insight and even look for drugs or compounds that might have an effect on these genes. All columns on this tab can be sorted and filtered to provide you with the information you need. You can choose to up the specificity and only look at those genes that changed in at least half the biosets, or you can decide you're only interested in, say, analyzing those genes that went down with infection. The possibilities are literally endless. If you still have questions on this data set, put them in the comment section for this video and I will try to answer them there. This concludes part 3 of this video series. In my next set of videos, we will get into the interpretation of the data set, which is by far my favorite part. I'll introduce an analytical program called ClueCore, which will allow us to visualize these data in 3-dimensional plots as well as clustered heat maps that will give us more information on our system. Stay safe, and I'll see you in the next video. Take care.